Production funding for QED with Dr. B is provided by
Battelle cares about STEM because it was part of our founding. Gordon Battelle, our founder, believed in building an institute that would actually better society through the use of science and technology. And this year we're going to hit a goal of 1 million students a year that we'll be able to impact around the country.
American Electric Power Foundation, boundless energy for brighter futures. And by viewers like you. Thank you.
I'm Dr. Frederick Bertley, scientist and President and CEO of COSI. Welcome to QED with me, Dr. B. Let's talk about science.
Today we're going to talk about nanotechnology, which is technology built at the nanoscale. So Dr. B, how small is the nanoscale?
Nanoscales are really small. Think of your hand. You can look at your hand and it kind of looks flat. But if you have a magnifying glass, you can actually see their ridges. Then if you take a microscope and look at your skin cells, you start to see the cells come alive. Cells, bacteria, that's on the microscope or microscopy level. Nanotechnology is 1,000 times smaller than that, 1 × 10⁻⁹. And so you're talking about, like, your hair, for example, might be 50,000 nanometers in thickness. A piece of paper could be 100,000 nanometers. In fact, there's 7.8 billion people on planet Earth. If each of us were 1 nanometer, we can fit the entire world's population inside one of these Hot Wheel cars. How cool is that, and how small is that?
Later on in the show, we'll look at how nanotechnology can be used to listen in on conversations between bacteria. And then we'll talk about how experts are using nano-engineered clothing as a way to track all sorts of information about our health. But first, Dr. B, you got to visit with a nano-engineer who actually builds structures at the nanoscale.
Yeah, he's an expert on something called DNA origami. Now, normally when we think of origami, we're talking about folding pieces of paper into little boats, little cranes, but his lab's not folding paper, he's actually folding DNA, and that's super cool. We're talking about Dr. Michael Hudubah from Audubon University in Westerville, Central Ohio. Dr. Hudubah, so good to see you.
Dr. B, welcome to The Point at Audubon University. Let's take a walk.
You got it, you got it. So listen, so you're an engineer and you build devices at the nano scale, but you use DNA technology. Tell us about that, and tell us about this thing called DNA origami.
When people think of, oh, you do DNA nanotechnology, they always think of genes and things like that, right? The stuff that make you, you, and me. But to us, in DNA origami, DNA is our brick and mortar. It's our steel, it's our cement. It is literally the material that we make machines out of. The other good thing about DNA is it actually assembles itself. There are 4 what we call nucleotides to DNA, A, T, G, and C. And the rule is simple. A binds with T, C binds with G. And so it's almost like a puzzle piece. What we do is we provide the pieces and provide the blueprint of what we want the puzzle to look like. But then if we mix all of those puzzle pieces together, put it under the right conditions like temperature and salt, the puzzle will actually form itself. The scaffold is going to fold into our predetermined shape, and that's why we call it DNA origami.
Are there therapeutic uses or applications for this?
Absolutely. So colleagues of mine at The Ohio State University have developed a structure that they call the horse. It's called a horse because of the Trojan horse, right? The story of the Trojan horse. So a lot of times, cancerous cells know when we're trying to give it cancerous drugs. And so before those drugs can get to the nucleus and do their dirty work, they're actually spit out from the cell membrane. So what they've done is actually hidden the cancerous drug inside of DNA origami nanomachines. So they're absorbed by the cell, absorbed by the nucleus, they disintegrate in the nucleus and release their payload.
That is so cool.
So that's not even the most exciting part. Dr. B, I got a surprise for you.
What?
Right now, I have a setup in the lab, so you get to go and make your very own DNA origami Trojan horse.
Are you kidding me?
I am not.
That's awesome. Let's do this.
All right. All right, so we're here. We're gonna make some DNA origami. We're gonna make a nanostructure called the horse. You're gonna mix all of our ingredients and put them in our water bath so we can fold this nanostructure DNA origami consists of two primary ingredients, the DNA scaffold and the DNA staples. This first ingredient you're adding is what we call the scaffold. So the scaffold is the major foundation of the structure. Think of it as the scaffolding of this building. All right, the next ingredient is called the folding reaction buffer. All right, this third ingredient is the salt. Our fourth ingredient here, we call this the working stock. So this is all of the other DNA staples. There's hundreds and hundreds of staples in there that are going to anneal with the scaffold to ultimately make the structure. So that's when you have your two single-stranded pieces of DNA separately. Annealing is when they come together to make that double helix.
Got it. It's kind of like two pieces of Velcro coming together.
Exactly. So let's go ahead and mix this up. All right, and so now what we want to do is we're going to put it in our water bath between 65 and 70 degrees. For 5 minutes, we're going to melt it at a really high temperature. And then what's going to happen is we're going to put it in what we call the folding temperature, which we've experimentally determined to be 52.5 degrees. Okay. We're going to put it in there for 10 minutes, and that's when all these pieces are going to very nicely anneal, but fold together, again, origami, and make our nanostructure. All right, so now we're going to load our structures into the gels. What we're going to do is basically put what you made into these little pockets in this jelly-like thing that we've created. Right? And DNA is negatively charged, so we're going to apply a voltage across here, and what's going to happen is that negatively charged DNA is going to go towards the positive end of that voltage, and we're going to be able to see it as it travels through. So what this does for us, things that are nice and compact, like our well-folded origami structure, are going to travel faster and all the same speeds. So we're actually going to not only see our structures travel through, but this is also purifying it, because anything that didn't fold well is going to be caught in the web, and everything that's nice and tightly packed is going to streamline its way through. That green light tells us that we're now applying our voltage, and now we wait 40 minutes.
All right, you know I'm nervous.
All right, so what I'm going to let you do the honors. What we should be looking for here is our right four bands. Do we see some nice, distinct bands of light?
Oh, yes. Yes.
You have made a DNA origami Trojan horse. You have made a machine out of DNA.
That is truly amazing. So not only are you a great teacher, I heard that you have a Guinness Book World Record.
I do. I was giving a presentation in Germany, and at the end I put up a picture of the Script Ohio that the Ohio State Marching Band does, and asked if there was any questions. And one of the questions I got was, why don't you make that out of DNA? And it was a brilliant idea, and almost 3 years later, I created one, it worked. And it's the smallest logo made with an uninterrupted strand of DNA in the history of mankind.
What is the future of nanotechnology and these devices?
Yeah, I think the best answer to that question is I don't know. You know, that's how science works. Nanotechnology was created in the early 80s. DNA origami was in 2006. We're creating the basic tools, the basic building blocks. But one thing I am really excited about with DNA nanotechnology is actually digital data storage. So just like computer code is zeros and ones, DNA is A's, C's, G's, and T's. It's just a sequence. But it has the ability to store information at a density of about 1 billion times more than what we can do today. And it doesn't need electricity or heat like a transistor does.
Dr. Hudubah, thank you so much for spending time with us.
It was my pleasure.
I love that he built that Script Ohio out of DNA, and to think that maybe one day we can use DNA for data storage, that's so futuristic. We know that nanotechnology can build things, but our next guest actually uses nanotechnology to spy on bacteria. Is that some sort of super cool James Bond spy device?
Definitely something like that. This group of researchers is actually studying bacterial infections and seeing how they can prevent them before they start. But to do that, they do it by listening to them. That's right, bacteria communicate just like we do. And for more on that, I connected with Dr. Fatima Alzaraz Alatratsky, who's a nanophysicist and researcher at Roskilde University in Denmark. But you actually spend a lot of your time studying bacteria, which are larger than nanotechnology. But I think you work in an aspect of bacteria that actually does work at that level. Can you talk about what you work in and why it'd be interesting to have you on a nanotechnology show?
So my work is very interdisciplinary. We work in between nanotechnology and the bacterial world. And the thing about the bacterial communication with which I work mostly within is that it happens on a nanoscale. Many of these molecules that are secreted to communicate, that the cells use to communicate with each other, are produced in nano levels. So it's nanomolar concentrations, and it's so small concentrations that it's amazing that it makes such a huge effect on us as hosts.
How do bacteria actually communicate? How does that work? And then what are they saying?
What they do is that they send signaling molecules out in the extracellular space. So it's all about sending out signaling molecules, receiving them, and then sending the response back. And all of this, the signaling out and the response, all of these molecules are called quorum sensing molecules.
Quorum sensing. Okay, so what do we mean by quorum sensing by bacteria?
So quorum actually comes from this ancient history of, or also from the business world, that we have a minimum person number that can take a decision in a meeting, for instance. And this has been adapted in microbiology, and more specifically in what is called social microbiology, where bacteria are able to decide based on not how many they are, but indirectly by the density of the quorum sensing molecules, these signaling molecules that are secreted in their space.
How are you essentially using the tools of nanotechnology to spy on the bacteria in the human body?
So we place artificial receptors or nanostructures that look like receptors. And in this way, whenever we have a sample with some signaling molecules, we would be able to capture these molecules that the bacteria were sending to each other, but in this way we capture them first.
What do we actually know from bacteria communicating?
The first thing I learned is that they are extremely complex. So this whole process of communicating, I thought in the beginning that it would be easy, like, you know, just like our language. I say something, you respond with something, but with bacteria, it's really depending on the context. We need to first of all understand when do they secrete something and in what context they secrete it and what are these molecules and in what quantities. And when we have understood that, then we are able to kind of put it all together like a puzzle and say, okay, in the beginning of an infection, they secrete this, they do it in these amounts, they do it differently if they're exposed to antibiotics, for example, and how do they do it then when it's late in the infection. And in this way, when we put the puzzle together, this is the work we can use for our benefit as humans.
Can you share an example of how bacteria communicate?
So one example is that within this quorum sensing system is that they can produce molecules for the public good of their local society. And in this way, they are able to collaborate. They just produce signals that don't just benefit themselves, but everyone in the community. And what happens sometimes is some cells figure out that if they don't produce, then they can just scout on everyone else's production. And what happens is that sometimes many cells can do that. They cheat. And when many cells cheat, we call it a cheater invasion. And this is a very interesting phenomena because sometimes they end up like having a community with a lot of cheaters, and that just collapses the community. So just thinking about the fact that in many ways they operate like us humans. They need to take care of the common good of their community for them to survive as a whole.
So by listening to bacteria and understanding how to communicate and understanding their conversations, we can actually translate that into advances in medicine and help us treat bacteria or deal with these infections?
We have a long way ahead of us to be able to translate everything and understand everything. But there are also other ways we can use this. I mean, just because they are able to communicate and to send these signaling molecules to each other, imagine that we are able to make them unable to recognize these molecules. Like if they're not able to listen to each other, then how will they communicate and how will they coordinate their activities? So this is a way to find alternatives to antibiotics.
So you're part microbiologist, part molecular biologist, part nanotechnology expert, and part code cracker? Like you're actually cracking a language.
We hope so. And it's, of course, a collaborative work. So I work with a lot of people in different fields. I have a great collaboration with medical doctors from where we get samples from real patients and we follow how is the infection progressing.
So you're working on a technology that could one day allow the everyday person insight into these bacterial communications.
So the big perspective here is to be able to translate every single secretion into this intracellular space and be able to translate it into actions. So if we somehow are able to use a smartphone or combine it with these nanosensors and detect on the spot, we will be able to, first of all, diagnose early, and we will also be able to figure out what treatment is necessary here and tailor this treatment to the specific patient.
It's amazing that soon doctors could be using a cell phone app to spy on bacteria conversations. You know, nanotechnology really is a big part of our day-to-day lives. So Dr. B, are there any other sort of creative applications for nanotechnology?
Diana, you're so right. There are many applications, everything from food safety to making fuel cells more efficient to even medicines helping to take care of tumors and other diseases within our body. But what I'm most excited about is how nanotechnology impacts our daily lives. And I'm talking specifically about the clothes and fashion industry and the stuff that we wear every day, making us look so cool. And for more on that, I talked with Dr. Juan Hinestroza, the Rebecca Q. Morgan Professor of Fiber Science and Apparel Design at Cornell University. He's an expert on nanomaterials and textiles. I don't know that many of us really think about the clothes we wear as full of science, but you refer to clothing as technology. Can you explain that?
Yeah, clothing is an incredible technological platform that is attached to our bodies, and very few things are actually attached to our bodies for long periods of time. You wear clothes for several hours, so you can get a lot of information about the clothing, your environment, your body, but also from the outside, where you are located. So maybe sensors to detect the quality of air, the amount of UV you are exposed to, the things that you're eating, how your body reacts, the sweat. Clothing, you can get the entire story of what happened on your entire day, because we can leave our home without a phone, yeah, but we cannot leave our home naked. I can guarantee you that. Clothes will always be next to you.
So in the future, and probably not so distant future, our jeans and t-shirts might be wearable sensors that can tell us if we're having kidney failure, heart issues, etc.
That is actually a reality in many cases, especially for military applications and also for people in space, where you can monitor remotely how they are feeling, how they are reacting to different environments and conditions. We are a moving platform. Imagine we can detect, for example, pollution in one area or exposure to different chemicals and communicate that information.
As a chemical engineer, what brought you into this unique intersection of textiles and nanotechnology? Now I hear superheroes might be involved.
Most of the superheroes, the power comes from clothing. Superman's cape that allows you to fly, to stop the bullet. Batman stops the fire. Aquaman can go into big depths in the ocean without changing the shape. Flash can go extremely fast with low friction between the air and the clothing. So I'm fascinated by these textile-enabled superpowers. Yeah, also fascinating because they were developed in the 1930s and 1940s when these materials did not exist. But somebody had the imagination that these materials maybe one day will exist, and that inspired a lot of chemists like me. Luckily, 40 years later, all these materials do exist. We use them every day. You see them in NASCAR clothing that prevents fire. You see with your firefighters in your neighborhood. In the Olympics, with this fancy clothing that can make you very fast in the swimming pool. I'm fascinated by that transformation and the power that textiles as a platform can enable. Basically, make us all superheroes.
Do you have a separate favorite project or another favorite project you've worked on that merges science and design?
Yeah, so I'm very lucky because in my department we have fashion designers and we also have architects and painters. A few years ago, we worked with a student from Africa, and she was fascinated by some of the molecules that we were developing. And she wanted to use these molecules to develop a new mosquito bed net to kill mosquitoes that transmit malaria. So we designed a molecule just for that, and then she designed an entire collection based on that molecule and in that particular technology. So what we do is we work with these molecules that are fascinating, are called metal organic frameworks. So basically it's like a cage. You imagine a cube, for example. All the points in the cube are actual metals and they are connected through an organic layer. So we made these layers to be attached to the cotton, and we grow the molecule from inside the cotton. So we use cotton as a platform to grow. Instead of attaching to, we grow like a building. So a building usually has a foundation, and then you make the building from the foundation up. We do the same atom by atom. So in that way, we can build the framework or our building. And then inside this framework, we have the insecticide. And this is the insecticide that will be released at the time that the mosquito is more active. So we basically calculate based on the amount of UV radiation, when do you open or close? Yeah. So maybe the mosquito will come between 5 and 7. Probably this is the time we're gonna open. The other mosquito will come about 4 p.m. where lights. So we can actually tune that to the scheduling. Basically, we want to match the schedule of the mosquito. Yeah. So look into his calendar and match it with us. So that was a good example because it affected a real life problem, and the family of this person have experienced malaria. So it's not like an imaginary problem. The big complex problems cannot be solved only by scientists or only by designers. You have to work together because complex problems require complex solutions. And I love the way these ways of thinking are so complex and different that the solution is usually at the interface.
What would you want our viewers to leave knowing about your research?
I would say that everywhere you look at, you can see science, and you can see the power of ingenuity and the power of science. From the moment that you are sleeping on your pillow, those are textiles. When you brush your teeth, that's another fiber. You can get information from brushing your teeth, from the food that you eat that has fibers as well as cellulose, the clothes that you have every day, the napkins that you clean your mouth after you're eating. All these fibers have an incredible amount of science. And if we look closely and deeply, we can see how we can use these items that we take for granted every day.
So my takeaway today is that the potential of nanotechnology is amazing, and it really feels like the sky is the limit there. Dr. B, what about you?
You know, it's a really interesting field because yes, we can start to understand how bacteria and even viruses communicate, but then understanding how everything is made up of small molecules at the atomic level and learning how to manipulate those to make small motors that are microscopic or on the nanoscale, from making medicines that you can't see with your eyes or a microscope, but to take care of diseases, to a whole bunch of other material science applications that'll make computers faster, make our cars safer. Just understanding this nanoscale, the science of the really, really small is transformational and is clearly going to help us on the planet. And that's QED with Dr. B. That's me. Find us on Facebook, Instagram, and Twitter, and we'll see you next time. Even though nanotechnology as a term gained traction in the 70s and 80s, the use of nanoparticles has been around for centuries. We just didn't know what the science was behind it. Did you know some of the most spectacular stained glass windows from the medieval times are the result of nanotechnology? By adding gold chloride and silver nitrate to molten glass, artists could obtain beautiful reds and yellows. They enjoyed the colors then, and we do now, because of how the glass and these substances reacted at the nano level. There's some really fun facts in nanotechnology. Did you know that 1 nanometer is about as long as your fingernail grows in 1 second? That's pretty cool. That was a second. It grew another nanometer, grew another 2 nanometers. Oh my gosh, 10 nanometers long. This is unbelievable. When we measure in nanometers, we get some wildly huge numbers. For example, how wide do you think the average human hair is? Is it 10,000 nanometers, 50,000 nanometers, or 90,000 nanometers? Now, if you're listening to the beginning of the show, you would have heard the answer. If you guessed 50,000 nanometers, you're in the ballpark. That's about the thickness of a human hair. And think about it, if you had 2 or 3 of those, like I wish I had on my head, that would be tons and tons of nanometers on your head. Pretty funny, right? So this is called a micropipetter. It is the tool for any molecular biologist. Kind of like your laptop, your pencil, your screwdriver, hammer. This is what every molecular biologist wants and has to use. And what it does is it allows you to get really small volumes in the quantities of what they call microliters. So we're gonna get 5 microliters. All right, to do that, you take one of these things. This is called the pipette tip. So here I'm opening this little tube, taking out the small amount of liquid, and then I'm gonna add it to this other tube here. And then I get to do the funnest part, is to mix it with this mini mixer or vortex. And when you push down, it rotates. And now your mixture is mixed.
Production funding for QED with Dr. B is provided by
Battelle cares about STEM because it was part of our founding. Gordon Battelle, our founder, believed in building an institute that would actually better society through the use of science and technology. And this year we're going to hit a goal of 1 million students a year that we'll be able to impact around the country.
American Electric Power Foundation, boundless energy for brighter futures. And by viewers like you. Thank you.